Hi everyone, welcome to the third lecture of urinary, and here we're going to be talking a little bit more about the processing of filtrate, which we began in the first and second lecture, and we're going to continue that today. Pretty much we're going to wrap it up. We will go a little bit more detail in the next two lectures, but this is really going to be the last of the big filtrate processing lectures. So let's go ahead and talk about our not really attendance questions for the day. Where does sodium first appear in the filtrate? What is the greatest pressure in glomerular filtration? And which part of the kidneys secrete renin? So go ahead and pause your video and try to answer those questions. And let's see what the correct answer should be. Where does sodium first appear in the filtrate? Well, sodium is something that is filtered, so sodium first appears in the filtrate inside Bowman's capsule. Once we move into the rest of the nephron, the sodium is already there. What is the greatest pressure in glomerular filtration? This is that outward hydrostatic pressure that's in the in the glomerulus once we come in from the afferent arteriole. So this pressure is the hydrostatic pressure of the glomerulus. That is the highest of those pressures. Which part of the kidney secretes renin? Well, back when we were first talking about the renin-angiotensin mechanism, way back in the first unit, we just said that the kidneys secrete renin when there's a drop in blood pressure. But now we have to be more specific. It is the JG cells specifically. Remember, they were also called granular cells, but we have to be that specific now and say the JG cells, the juxtaglomerular cells. Remember, granular cells are all over the body, and the kidneys, we give them a special name. So it is the JG cells that secrete renin.
Today's lecture is going to be just kind of a very broad overview of reabsorption, secretion. We're going to talk mostly about the proximal convoluted tubule and the ways that substances are reabsorbed. Next lecture, we will wrap up reabsorption and secretion along the rest of the nephron. And then the third lecture this week, so the fifth lecture of this online chapter, will be over something called the countercurrent mechanism, which really focuses in on the loop of Henle. So let's dive in and start talking about reabsorption, and to a small degree, maybe we'll talk a little bit about secretion. Again, here's our more realistic view of nephrons, and now the reason that we're going to talk about that is because when we look at the nephron here, which is a little like that generalized nephron that we looked at, we can see the tubules, but it leaves out the fact that these purple guys right here, these are capillaries. So if we are reabsorbing or secreting substances all along these tubules, remember, reabsorbing is taking something out of the filtrate in these tubules and putting it into the blood. Secretion is taking stuff from the blood and putting it into the filtrate in these tubules. So we want the blood to be very, very close by. And that's why this layout is important to know. The entire nephron is surrounded by two different types of capillaries. We've got paratubular capillaries, which surround every bit of a cortical nephron, and the PCT and the DCT of a juxtamedullary nephron. And then we also have a special capillary called the vasa recta, which surrounds the loop of Henle in juxtamedullary nephrons. Make sure if you didn't get the difference between juxtamedullary and cortical nephrons, you clear that up, because next class we're going to really focus on the juxtamedullary nephrons. That actually came from the self-study portion. But we can see not only are these capillaries completely surrounding the nephrons, there's actually a little bit of space in between the tubules and the nephrons. So when we are, for example, reabsorbing something, yes, it does go from the filtrate that's inside the tubules into the blood. But a better way to say that, a more realistic way to say that, is substances move from the filtrate inside the tubules into the interstitium, the area just in between cells. And then from the interstitium, it moves into the blood. So for example, if you see my pointer here, let's just look right here. If something is reabsorbed, it's going to move from the filtrate inside the tubule right here into the interstitium, and then from the interstitium into the capillary blood. In secretion, something would move from the capillary blood into the interstitium, and then from the interstitium into the tubule containing the filtrate. So it does go the direction that we've said, it's just there is an intermediate step as that substance passes through the interstitium. So, first looking at the proximal convoluted tubule. This is as soon as that filtrate leaves Bowman's capsule and it moves into the first part of the tubule, the proximal convoluted tubule, the PCT. And here in the PCT, about 65% of the filtrate that we just made inside Bowman's capsule is immediately reabsorbed. So over half of the filtrate that we produce is reabsorbed in the very first part of the kidney tubules. About 6% of the calorie intake per day powers the processes of the PCT. There is a lot going on in the PCT to reclaim all of those substances that our body needs to hold on to. It uses a lot of energy. And when we talk about reabsorption, we're going to talk about transcellular processes and paracellular processes. Transcellular, something is moving from the lumen, the hollow part of the tubule, where the filtrate is, into a cell on one side, through the entire cell, and out the cell on the other side. So the tubule is made of cells, like all of the parts of our body. It's made of cells. So it is a single layer thick. The tubules are made of simple squamous or simple cuboidal, or in some cases, simple columnar epithelium. And as that substance moves through, it's going to pass through that single layer of cells. That's the transcellular process. In the paracellular process, it's a substance moving between those cells. So it's not passing through a cell, it's passing in the space between those cells. And different substances will do different methods. So first let's look at sodium, because sodium is really going to be the key player for practically everything we talk about the rest of this chapter. Sodium creates an osmotic gradient. Think back to what we've said all semester long, solutes suck. And when we talk about solutes, the primary solute in our body is sodium. Sodium creates osmotic gradients, and what that means is wherever sodium goes, it pulls water towards itself. Another way to say that is where sodium goes, water follows. It just pulls the water along with it. So if we reabsorb sodium, it will also pull water along with it. We saw that in the renin-angiotensin mechanism that we drew at the end of the last lecture. When aldosterone was released, it created those sodium-potassium pumps to reabsorb sodium. ADH installed those aquaporins that let water get pulled through. Where was the water going? It was going towards the sodium that we just reabsorbed. So it's all about reabsorbing sodium. Now we're going to introduce two new terms, the basal side and the apical side. And that's referring to those cells that create the walls of those kidney tubules. The basal side is the side closest to the blood vessels. It's facing the interstitium. So let's imagine here. We can see here's the PCT, we've taken a bit of it, and now we're looking at these are the cells that make the wall of the tubule. Here it is a simple squamous epithelial, a simple cuboidal epithelial cell in this case. Over here is the filtrate of the tubule inside the lumen. Here is the interstitium that we just mentioned. And here is one of those peritubular capillaries. So when we reabsorb something, something is passing from the filtrate and then either through the transcellular route or through the paracellular route between the cells. But as we just saw here, the basal side of that cell is the cell closest to the blood vessel, the interstitium. This is the basal side of that cell. The apical side is the side closest to the lumen of the tubule, closest to the inside of the tubule where the filtrate is. So apical side, basal side. So let's talk about the basal side first. The activities going on the basal side of the cells and the PCT regarding sodium. There's a lot going on both sides. Right now we're just concerned about what's going on regarding sodium. So we have primary active transport, those sodium-potassium pumps. Now whether or not aldosterone is present, we're always going to have some sodium-potassium pumps. We saw last class that aldosterone just makes more sodium-potassium pumps, but it actually makes those much later in the nephron. It makes those in the DCT and the collecting duct. So we're in the PCT right now. On the basal side, we have primary active transport, the sodium-potassium pump, which is what we see right here. The sodium-potassium pump uses ATP, and it's constantly moving sodium, 3 of them, out of the cell and 2 potassiums into the cell. Constantly. 3 sodiums out, 2 potassiums in. 3 more sodium out, 2 more potassium in. Over and over and over. Now remember, potassium is always leaking out of every cell in our body down its gradient. So down its electrical gradient. Down its concentration gradient. It leaks in through its electrical gradient, but out through its concentration gradient. So potassium, once it's pumped in, it leaks out. And then we pump it back in, and it leaks back out. We pump it back in, it leaks back out. That seems kind of, counterproductive. Why do we do that? Well, we're not doing it because of potassium. We're doing it so that we can be moving sodium out of this cell constantly. The potassium just kind of gets recycled. Some of it does move into the bloodstream to get carried away, but more importantly, it's the sodium constantly getting moved out of the cell. Why do we want to move the sodium out of the cell? To create an osmotic gradient. All right, so we'll come back to that. Now let's go back and look at the apical side, that side that's closest to the lumen of the tubule, facing where the filtrate is. Now that basal activity is creating an electrochemical gradient for sodium. Let's go back and look at what that means. We're pumping all this sodium out. So not only are we lowering the sodium concentration, we are also pumping 3 positives out, but only 2 positives are coming in. 3 positives out, only 2 positives coming in. So we're losing positive charges every time we do that. We're making the inside of the cell more negative, just like every cell in our body. Inside of the cell is more negative. That is then going to draw sodium into the cell on the apical side. And sodium's going to come in a few ways. It's going to come in through things called symports. So that a symport is something that transports 2 or more things at the same time. So, for example, sodium and glucose, sodium and amino acid of some particular amino acid, sodium and certain vitamins, particular vitamins. So sodium plus something. Why is the sodium coming in with one of these items? Because of the negative charge and the low concentration of sodium. Why do we have a negative charge and a low concentration of sodium? Because the sodium-potassium pump keeps pushing those sodiums out. We push sodiums out, low concentration of sodium pulls more sodiums in on this side. Pump more sodiums out, keep the concentration low, also keep the inside more negative. If the inside is more negative, sodium, with its positive charge, wants to come in like a magnet. As soon as it comes in on this side, it gets pumped out on the other side. So it's like a conveyor belt. We're constantly removing sodium on this side, which allows sodium to come in along with some friends on the other side. Not only are we reabsorbing sodium, which has an osmotic gradient, wanting to pull water towards itself, it also is causing us to reabsorb other nutrients, glucose, amino acids, vitamins, things like that, because they come in along with the sodium that's being drawn in to the negative concentration, the negative charge, and the low concentration inside the cell. As soon as that sodium comes in, it goes right across, out the sodium-potassium pump, into the interstitium, ultimately into the blood to be carried away. Also, we have things called sodium and hydrogen antiports. Antiports are the opposite of symports. They move two things, but they're moving them in opposite directions. It's moving sodium one way, and in this case, a hydrogen ion the other way. We don't have an example of that here, but we can see it a little bit later on another diagram. But what we have is sodium comes in and H plus goes into the filtrate, which allows actually hydrogen ions to move from the blood into the cell, ultimately into the filtrate. Everything here is all about reabsorbing sodium, and reabsorbing sodium so that we can reabsorb water. Also in the proximal convoluted tubule, we'll have chloride. Chloride is going to go wherever the sodium goes. It's got a negative charge. It is attracted to the positive charge of sodium. Antiports are on the apical side, and those on the basal side, we've got potassium chloride symporters. We'll see those again here in just a minute when we get to another diagram. But in this case, we're moving potassium and chloride in the same direction. In antiports, we are moving chloride and something in the opposite direction. We also have other electrolytes that are being reabsorbed. Potassium, magnesium, phosphate, through the paracellular pathway. So they are going between cells in addition to what we saw going through the cells already. So here, electrolytes, I believe we're probably all familiar with this term already. I think I introduced it back when we were talking about plasma, but just to make sure everyone knows, electrolytes are just things that develop a charge when they are in water. So all of those things like Na+, K+, Ca2+, Cl-, things that have a charge when they are in water are electrolytes. Phosphate and sodium are co-transported. So sodium going along and potassium coming along with it. Calcium, Ca2+. Calcium comes through the paracellular and the transcellular pathway. Glucose. So 100% of the glucose that was filtered at the glomerulus inside Bowman's capsule is reabsorbed in the PCT. That is in the average healthy adult male. Now, someone who has more than normal glucose, then we exceed that 100%. We'll see that in just a minute. But when someone has the normal amount of glucose in their blood, and that glucose is filtered at the glomerulus, every bit of it is reabsorbed in the PCT. It goes right back into our blood because glucose is our source of energy for making ATP. Now, we have sodium glucose transporters on the apical side. Then we have channels on the basal side. So we're moving glucose into the cell on the side closest to the filtrate. The glucose comes in with sodium. As it passes through the cell on the basal side, the side facing the blood vessels, there are just channels that allow glucose to pass through into the interstitium and ultimately into the blood to be carried away. So we've been talking a lot about sodium, and we keep saying that sodium is being reabsorbed so that we can reabsorb water. So now let's look at the water side of it, because really this is what we're after in the end. It's water that we're trying to reabsorb. And when we reabsorb water, water is going to pass through these small channels in our cells called aquaporins. Water is reabsorbed through aquaporins. And let's jump down here to the bottom first, because I want to go ahead and talk about what an aquaporin is. Aquaporins, like I just said, they're channels that water can pass through. And there's two different types. There's something called aquaporin type 1, and those aquaporins are always there. They are always there regardless, and water is constantly being reabsorbed through them. Aquaporin type 2 is only present when ADH is being secreted by our hypothalamus. So when we release ADH, it tells our kidney tubules to make aquaporin type 2. Now we have more aquaporins in our kidney tubules, which allows us to reabsorb more water. And remember, that's what ADH does. It tells our kidneys to reabsorb more water. Now we see the mechanism that happens through. ADH makes aquaporin type 2 in our kidney tubules. Now we have more than normal aquaporins, so we can reabsorb more than the normal amount of water. How much water do we reabsorb? Well, 180 liters of filtrate produces 1 to 2 liters of urine daily. So what happens to all of that other filtrate? Most of it is water, and most of it is reabsorbed. By far and away, most of that water is reabsorbed. We went from 180 liters, that was mostly water, to 1 to 2 liters, that is mostly water. And 2/3 of that water that was reabsorbed is reabsorbed at the PCT. So the PCT is reabsorbing the bulk of the water that we reabsorb. And it's due to osmosis. What's the osmotic force that's causing this osmosis? Those solutes that we reabsorbed, primarily sodium. Reabsorbed solutes, water will come along through those type 1 aquaporins, and when ADH is present, it will also come along through those type 2 aquaporins. Reabsorb solutes, then we can reabsorb the water through osmosis. So again, here's that same picture that we just looked at. We're reabsorbing these solutes, which allows water to come through. And right here, what do we see? This is an aquaporin. Here's another aquaporin on the other side. Water is coming through, trying to get to those solutes that we just reabsorbed. Now here is the one that's actually in your book. This is on page 902 of your textbook. It's flipped on the other side. Over here is the tubule, here's the cell, here's the interstitium, and here's the capillary. And this shows more of those transporters, the symports, the antiports, that we've been talking about. So you can look through this and see the ones that we've mentioned so far. But I want to talk about transport maximum. And this will be the last thing that we talk about today, but it's important to see exactly how and why it's important to understand those mechanisms that we're reabsorbing things. So let's look at glucose as an example. And what we say with glucose can also be applied to amino acids, can also be applied to vitamins, can also be applied to various substances that are only reabsorbed in the PCT especially. You can apply it to anything in the nephron at all, but glucose, amino acids, and vitamins are only reabsorbed in the PCT, so it's very important to talk about transport maximum with regards to those. When something is moving through the nephron, let's be a little more specific. When glucose is moving through the PCT, it's being reabsorbed. How's it being reabsorbed? It's being moved through proteins that allow glucose to pass through. We could say the same thing for a specific amino acid. We could say the same thing for a specific vitamin. We're just using glucose in our example here. So up here in this picture, these blue dots, let's say they represent glucose. And these little blue circles, these represent the carriers that are allowing glucose to pass through. And they're only found in the PCT when we're talking about glucose, amino acids, and vitamins. So in someone with a normal amount of glucose in their blood, that glucose is filtered, and as that glucose passes through the PCT, these proteins grab the glucose and move it out so that it can ultimately make its way back into our bloodstream. And by the time we get to the end of the PCT, we have reabsorbed 100% of that glucose. And there's a rate at which these work. If we have too much glucose, let's see what happens. Over here, let's say this is someone with hyperglycemia, someone with high blood sugar. Maybe it's someone with diabetes mellitus, which is the type of diabetes most of us are familiar with. So here, now we see there's a lot more glucose in the blood, so there's going to be a lot more glucose in the filtrate. As that glucose passes through the PCT, then these proteins are grabbing the glucose and reabsorbing it, grabbing the glucose and reabsorbing it. But by the time we get to the end of the PCT, there's still a lot of glucose left. And from the end of the PCT through the rest of the nephron, there are no more glucose transport proteins. Same with amino acids, same with vitamins. Once you get to the end of the PCT, there are no more transport proteins for them. So, how much glucose can each of these carry, or how much amino acids can each of these carry? They all have a rate, and that rate is called the transport maximum. And if we have more of the substance, if we exceed transport maximum, what happens? Well, that substance remains in the filtrate. And if it remains in the filtrate, it's also going to show up in the urine. There should not be glucose in the urine normally because we've reabsorbed it all. Down here we see there's no glucose in the urine. Over here we exceeded transport maximum. Now there's glucose in the urine. But there's another effect, something called osmotic diuresis. So I'm going to backtrack and get our picture of the nephron here. Here's our PCT. As we move through normally, glucose is all reabsorbed by the time we get to right here. But if we exceed transport maximum, if there's too much glucose, then from this point on, there will still be glucose in the urine. All right, now, or in the filtrate that shows up in the urine. But let's look at what else happens. As we pass through the descending limb of the loop of Henle, we're reabsorbing water. As we pass through this DCT, we're reabsorbing water. As we pass through the collecting duct, we're reabsorbing water. But if there's more glucose in the urine, or if there's more amino acids in the urine, or if there's more vitamins in the urine, what happens? Well, those are solutes. What do solutes do? They suck. So as we pass through the descending limb of Leukopenly, yeah, we're going to reabsorb some water, but we're not going to reabsorb as much as normal because that glucose is going to hold on to some of that water. When we get to the DCT, we're still going to reabsorb some water, but we're not going to reabsorb as much water because the glucose is going to hold on to it, again in the collecting duct. So by the time we get to the end of the collecting duct, that solute, glucose or amino acids or vitamins, they have pulled a lot more water along with them than we would normally expect to find. So not only do we see glucose in the urine, we see a lot of water in the urine. Another way to say that is it causes an increase in urine volume. When something exceeds transport maximum, one, that substance shows up in the urine, and two, there is an increase in urine volume. Let's tie this to something else that a lot of you are probably familiar with. If you've ever worked in a hospital or clinic setting, it's common practice to see people taking over-the-counter vitamins, or if they're in the hospital, they're given multivitamins. And really, honestly, for the most part, unless you have a deficiency of some sort, multivitamins are useless because we reabsorb 100% of the vitamins that was filtered. And if we have more than normal vitamins, what happens? It exceeds transport maximum and it shows up in the urine. That multivitamin that you just gave them, they just peed it away. And it's common to hear people that work in these settings, when you give someone a multivitamin, they say, we're going to give this patient expensive pee because you're charging them a lot for these vitamins and it's passing straight through them. Now, to kind of wrap this all up, I think this is going to be the last topic. Yeah, so to kind of wrap this all up, I want to show you guys a video that I always like to introduce when we talk about this. And some of you are going to be old enough that you know exactly what I'm talking about, and some of you maybe not, because the longer I teach, the fewer students know what I'm talking about. But there was this show called I Love Lucy. I'm sure everyone's heard of I Love Lucy. And if you have ever seen even a single episode of I Love Lucy, you've seen the clip that I'm about to show. And it's going to tie in directly to what we just talked about with transport maximum. So let's look at the video, and then we'll talk about transport maximum as it's playing. So here's Lucy and her friend, and they're working in a chocolate factory. And you've probably seen this clip. Some chocolates are going to start coming along, and it's Lucy and her friend's job to grab these chocolates and pack them up for delivery. And as they keep going, the chocolates start coming faster and faster and faster, and they're trying to keep up. And eventually, there's so many chocolates coming so fast that they can't keep up, and the chocolates continue on and shoot off the end. Or we could also say the chocolates have exceeded transport maximum. So let's take a look. Right now, everything's moving fine, so they're keeping up. But now things are going to start to go a little bit faster. But they're still able to keep up. But now things have started to really pick up. We've got more glucose than normal in the blood. They're starting to have some problems. We are exceeding transport maximum. And they're not able to keep up with their job because the chocolates are going way too fast. There's too many chocolates. The chocolates have exceeded transport maximum. And there they go. They've passed through the PCT, and that's it. So, transport maximum, how well can we reabsorb whatever substance it is? If we exceed transport maximum, one, the substance shows up in the urine, and two, the urine volume is increased. So next class, we're going to wrap up with reabsorption and secretion. The lecture after that, we're going to talk about the loop of Henle and the countercurrent mechanism. All right, I will talk to you guys later. Let me know if you have any questions.